growing number of studies show that various tissues in the aged mouse could be rejuvenated with a surgical procedure termed parabiosis. In parabiosis, two mice, a young and an aged mouse, are surgically connected to share a common blood circulation. And the Weiss-Curie lab has studied the brains of these mice extensively and found that indeed the aged mouse brain was, many of aspects of aging were reversed, such as neuronal plasticity, restoration of neuronal plasticity, or reduction of neuroinflammation. So in follow-up study, they went on to transfer plasma of young mice to aged mice and vice versa, from aged to young. And this enabled to study each one of the mice separately in cognitive tests, for example. And indeed they found that exposure or injections of young plasma resulted in better performance of these aged mice in memory and learning tests. So the conclusion from all of these experiments was that there's youthful factors in young blood that are lost with aging, while other more toxic proteins are accumulating in the aged blood. But finding the precise proteins and how they, the mechanisms of how they action on the brain has been extremely challenging, because the brain is secluded from the blood circulation through barriers that limit the transfer of proteins into the brain. So to target that challenge in the study that I presented, we took one step inwards across these barriers into the brain and performed transfers of cerebrospinal fluid from young to aged mice. And CSF is very rich with the proteins and other factors that are known to affect brain development and support brain function. And it's routinely used in the clinic as a proxy for brain health. And we know that from studies that measure these proteins, we know that they change dramatically with age, as well as the way the CSF flows within the brain. So in the study, we collected CSF from young mice and very slowly infused it to the lateral ventricles of aged mice, and then screened for the cellular process or the cell type that would be the most affected from this manipulation. And to our surprise, oligodendrocytes emerged as having the highest transcriptional signature following CSF infusions. So we then zoomed in on these cells and their function and found that indeed oligodendrocyte progenitors, or OPCs, they started proliferating and differentiating following young CSF, and this was in vivo in the hippocampus as well as in vitro. We also found that following CSF infusions, aged mice did better in recall of long-term memories. And this is specifically interesting because it was recently shown that this specific memory function relies on the formation of new myelin, which fine-tunes the synchrony of these memory networks. And we're currently screening for potential factors or proteins in the CSF that could induce these beneficial effects on oligodendrocytes. So I think that the good news is that these cells are still present in the aged brain, and given the right cues, their function could be restored. It was recently shown that by putting aged mice or rats on an intermittent fasting diet, or administering them with certain drugs such as metformin or clemastine, the basic function of oligodendrocytes was restored, as well as their reaction to a demyelinating injury. So I'm hopeful that all these findings will lead to a treatment or an intervention that could also potentially be relevant to multiple sclerosis patients.